And
Good morning to all. Hello, everyone joining today's webinar. I'm Dr. Pradeep Golani, and as the co-convener of this event, I'm delighted to welcome a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to a productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I am confident that today's session will uphold our tradition of excellence, providing valuable insights from our esteemed speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us today. We are gathered today to commemorate the 77th SPSR International Webinar on Drug Product Development and Industrial Approach, a distinguished event in the series hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, the National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of knowledge about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Dr. Saroj Kumar Rout, Formulation and Process Development Scientist, R&D, LNK International, New York, USA, as a distinguished speaker. Dr. Saroj Kumar Rout's expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussions and expand exploration of drug product development and industrial approach. We are grateful for his engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the National Secretary of SPSR and Managing Editor of both IJPSR and IJP. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. Now we will start with a short introduction to the guest profile. Dr. Saroj Kumar Rout is a distinguished formulation and process development scientist currently associated with LNK International, Hoppage, New York, USA. With a passion for science and industrial pharmaceutical technology, Dr. Rout has dedicated over 15 years to advancing the field of pharmaceutical drug product development. His commitment to bridging the gap between the academia and industry drives his collaborative efforts with university research programs, fostering innovation and knowledge sharing. Dr. Rout holds a PhD in pharmaceutics, with his research focusing on enhancing the critical quality attributes of high drug load formulations. His expertise spans the end to end development of drug products for various markets, including ANDAs for the US and EU, clinical drug immediate release, extended release, and sustained release dosage forms. He has extensive experience in platform technologies such as control release platforms, and hot melt extrusions. His technical acumen extend to granulation technologies, including wet granulation, dry granulation, via roller compaction, and spray granulation, as well as drying, compression, and coating technologies. Dr. Rout has successfully developed various dosage forms, including bilayer tablets, and has mastered both functional and non functional coating processes. His vast experience and technical expertise contribute significantly to solving complex challenges in drug product development, paving the way for innovative pharmaceutical solutions. Dr. Rao's dedication to advancing pharmaceutical science, coupled with his ability to translate scientific research into practical applications, makes him a vital contributor to the global pharmaceutical industry. His work continues to impact the development of effective and high quality drug products benefiting patients and healthcare systems worldwide. It is with great satisfaction and pride that we declare that we have exceeded 2,000 registrants globally, showcasing an impressive diversity of countries. Significant attendance is noted from nations including India, Algeria, Bangladesh, Brazil, Cameroon, Egypt, Ethiopia, Indonesia, Jamaica, Kenya, Malaysia, Morocco, Nepal, Nigeria, Oman, Pakistan, Philippines, Saudi Arabia, Sudan, UAE, USA, and Vietnam. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each participant who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Saroj Kumar Rout, with us. Welcome, sir. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, sir.
Yeah, thank you, Dr. Pradeep, and for the kind introduction. And uh, I'm very thankful receiving the invitation from SPSR. I'm very fortunate to participate and share my experience with fellow students and colleagues and pharmaceutical professionals. With I would like to share all my experience, whatever I gained. The objective will be to share the experience so that the grassroots level we can develop and we can enrich, we can educate the basic research, which is going to help. I'm very thankful to a novel initiative taken by Dr. Shashi Alok for his invitation. So now I'm going to present my screen.
Right, sir.
Yeah. So is my screen is visible now?
Right, sir. It's visible, sir. Perfect.
Yeah. So before we start, going deep into the presentation, that presentation will be a general topic. Is about the drug product development, where how we as an industrial professional approach a drug product development. So basic research and how it is going to, how we perceive, or as an industrial scientist, how we uh, uh, use those basic research in terms of industrial applications. So yeah, so our discussion point will be revolving around this general topic. So it we'll have a brief discussion about the different phase of drug development and role and responsibility of formulation scientists in a industry, and that type of drug product development based on the regulatory norms, how what type of drug we develop, what is the regulatory requirement for each. That will be a brief discussion. Then we'll discuss about the drug product development with respect to process. Then we'll see one case study with respect to drug substance impact. So this is the disclaimer. So this presentation is, I collected all the information from the public resource, and it is typically meant for the uh, education purpose, and uh, which the goal of connecting with people around university is this is my objective, or this is a mission where I'm trying to involve in the university research work, where when university students or a research scholar at the grassroots level, they will come to know, like, what is happening exactly in the industry. So the research will be a applied research, so we can leverage all the basic research work in the industry, and that's vice versa. It will be like, it is, they will see that how we are taking their datas, or how we are doing the literature search, how we are utilizing those basic research in the industrial applications. So my goal is, it's a small step where I'm taking towards uh, to minimize the gap between industry and university. Yeah. So basically, to start with the discussion, so we should know the few other terminology, saying that, what is that basic terminology for which we are going to use during the uh, daily research work. Like, the best, these are the basic terminology, just to have um clarity on those part. These are taken from the regulatory sites. We, or everybody knows that, what is the drug substance. So a drug substance is any substance that used to prevent or diagnose or treat or relieve symptoms or abnormal condition. When the drug substance along with inactive ingredient mixed up, so it forms a drug product. So the point here I highlighted is, so most of the thing, maybe you'll see the I would like to highlight here, like, everybody, please concentrate on those highlighted parts, though it looks like it's subjective, but we see that what is drug product, since the presentation is more like we are discussing about the drug products and all. So what we'll see is, so it means that when we go for a drug product manufacturing, we see active and there is inactive ingredients. So inactive ingredients in the subsequent slide, we'll see, like, when we say it's a inactive ingredient. So what does it mean by inactive ingredients? So we'll go with some um, uh, examples, and what are the future scope in the field of drug substance and inactive ingredients. So I was just discussing about the inactive ingredients. So as we say, like, the ingredients are inactive in nature. So are they really inactive? or what does it mean by inactive ingredients here? So that they are inactive with respect to the manufacturing process. So future scope, what I can expect, like is there all is there any excipient still not evaluated for its therapeutic potential? So these are few future scope with respect to inactive ingredients, and few scopes with respect to the drug substance. So the point is here, can we modify a drug substance to make it suitable for better viability? So that means, for example, we have a drug substance and we included, in the final drug product, we are including a bioenhancer or a surfactant, something we are including in the while designing the drug product. So is there any possibility while synthesizing the drug substance, can we co-process a drug molecule along with a bioenhancer there? So this is one of the area where researcher can uh, focus on. So where there is a large scope where that can be considered as a future scope for the drug substance. And can we modify, a drug molecule? If suppose a drug molecule is, acid or alkali degradation, means it is prone to acid or alkali degradation. Is there any possibility to prevent the drug during the synthesis or during the recrystallization of the drug substance? So those are the few questions, even if we can ask ourselves, or we can say this can be a future scope where we can spend some time. Research scholar can spend some time in doing that research, basic research. There is another point is like when as a drug product scientist, when we see a drug substance is hygroscopic, it is creating challenge during the processability of the molecule, during the manufacturing, or during the formula and process development. Is there any possibility we can modify the drug substance during its synthetic stage or while doing the recrystallization stage? So this slide is basically with respect to possibility or future scope where we can work on, or we can spend some time doing the basic research. This is for the university and also the industry people where we can look on this topic. So going to the normal discussion of type of drug product development based on the regulatory requirement. These are the common chapters, like with new drug application. So it is the development of a NCE molecule, new chemical entity molecule, where the brand company gets 5 years of exclusivity, marketing exclusivity, where they may have uh, drug substance patent or drug product patent. ANDA, abbreviated new drug application. So it's a section of 505J, where the difference between why I kept in a single slide is the difference between this one and this one is NDA and ANDA is we have to have bioequivalence study, where here in the NDA new drug application we used to do the bioavailability study, where it starts with, we'll see the different phases. I have a graphical presentation for that. So there we do the viability and preclinical studies and all those steps, where in the ANDAs, basically ANDA do not contain any clinical studies as required in the NDA molecule. So we'll have a discussion on this in the next subsequent slides. And 5052 is a hybrid NDA type of development, where we we when we say like it is 505(b)(2) or hybrid NDA is when we change the dosage form or strength or route of administration of existing dosage form. So that is 505(b)(2) type of development. So this is what we were saying, like this is just what we discuss. NDA is a 505(b)(1), where we get the marketing brand company gets the marketing exclusivity for 5 years for the new chemical entity. We'll have a discussion how it goes, how the drug substance, what is the different stage of substance, drug product development, and abbreviated a new drug application where we have, as we say, just now we discuss, we have to copy the RLD, or we have to manufacture a pharmaceutical equivalent drug as per the NDA molecule. So here when we say pharmaceutical equivalent, we have to have the bioequivalence studies for the ANDAs, and this is 505(b)(2), as we discussed before. This slide, it is more related to changing dosage form or strength, routes of administration. So these are the basic requirement, regulatory, if you're going for a US market or Europe market, US market. So they are looking for these requirements. Yeah. So this is what we discuss. For the NDA molecule, we have to have that viability study, and these are the common definitions I have taken from the regulatory sites, and the rate and extent to which the active ingredient or active moiety is absorbed from the drug product and become available at the site of drug action. So, and a bioequivalence is more like pharmaceutical equivalent with the brand product or reference listed drug or RLD. Yeah. So when we say a drug substance is synthesized, and in the second slide, I was discussing about what are the future scope in terms of in the field of drug substance, where we can say like when a drug substance during the synthesis of a drug substance, if they can see that what is the future of how it is going to go through different stage of drug product development. So we can still add those few characters, like we, as I said, like if the molecule is very hygroscopic and or acid labile or alkali degrading, so we can have some co-process with some of the excipients to overcome those challenges. But when we start from the synthetic stage to final drug product, or to develop a successful, stable, safe, effective doses form, that time if you'll see the success rate here, the success rate is very low. When we say it will go through preclinical or clinical studies, the success rate is extremely low, and it is nearly 1 out of 20 to 30,000 molecule to be synthesized. So here the discussion point is, like, if suppose one molecule out of 20,000 or 30,000 molecule is successful, so where this 19,999 molecule goes? So that's the point where during the second slide presentation I was saying we can still have a scope here, where I would suggest a research scholar, they can still see on those parts. And obviously this is since there is very less or low success rate, and there is a huge investment required from the company side or from the organization. So the process starting from synthetic phase to drug product manufacturing or approval, a successful development of product, it will become a critical. For that, the process designing the process, making the drug stable in a particular process, particular if suppose we are going with a certain drug and it is degrading during in the stomach pH, acidic pH, so we need to have some alkalizer or something we used to add in the process. So those are the things we can concentrate on during the synthetic phase also. That is my hypothesis. So if we can still work on those parts, that will be really helpful. We can minimize this failure rate, or we can increase the success rate to maybe another 50%, maybe two molecule out of 20,000 molecule. So the molecule, when it starts with a discovery phase, where we create different molecule with different functional group or different active moiety. Then it has to go through preclinical phase, most critical phase before the administration to human body. So we used to have this animal study in this, and where we decide a route of administration, what is the viability, all those stuffs. Then we go for the phase 3 clinical study. Clinical study, again, it has different steps. We'll see on that part. Then we, whatever document, whenever we develop a successful drug product with a particular manufacturing process, then FDA, for US market, FDA reviews all those data, starting from all those initial phase data to the clinical phase data. During the clinical phase 3, and if FDA reviews all those data and it gives the approval for the marketing. Then once it is goes to market, then we will have the post-marketing safety monitoring. Now here the pharmacovigilance roles, all those, and observing, monitoring the drug in the market. What is the impact on the patient population? So it is monitored through phase 5. Yeah. So just now we discussed about the slide. So the first page will be identifying potential candidate that will interact with target molecule, have potential to become a drug candidate. So this is the phase 1, and phase 2 is the preclinical. This is the most important phase where we do all those in vitro study and in vivo study, where we decide on the drug molecule, whether it will go with oral route or IV, which polymer is stable, what will be the solubility, which salt form it will go. So all those forms, route of administration, and viability in the animals. We do all this in the animal studies. In the phase 3, this starts the clinical disease, where we concentrate on safety and efficacy of the compound in human. This will be the first time in the first phase, or FIH, first in human, where phase 1 is the first in human phase, where we used to take 20 to 100 volunteers participants. So maybe this will be healthy volunteers, or if the drug is anti-cancer, so it will be on patients. So regulatory or FDA says that we have to prove here the drug is safe. Once it is proved here in the animal study the drug is safe, then we can go for this the human trial with less number of volunteers. So 2200 participants. Phase 2 will be phase 2. Again, it has two phase: phase 2A, phase 2B, where we see the effectiveness and all the side effects, or we do the proof of concept, saying that we are saying that this has this therapeutic effect and it is showing this therapeutic effect. So all those phase 1, phase 2 is a process where difference is here, the number of volunteers here, and here in the phase 1 you have 2200 participants, and but where in the phase 2 the number will increase to 100, up to several hundred volunteers. And phase 3, yes. So phase 3 is a state where a phase where we see that effectiveness and adverse reactions. So the volunteers, number of volunteers, it will be certain up to thousands or 3,000 participants with different population. with disease condition at different and at different doses form. So once this phase 3 completed, so all the clinical data will be compiled and it will be submitted for the drug product risk-based benefit analysis, where we see that safety and effectiveness it is proved, and drug product risk benefit analysis is proved. Then FDA for the US market, they gives the approval for the marketing marketing of the drug product. Once it is in market, as we say, when it is in market, so the post-marketing safety monitoring will be conducted. So this is basic of the drug product development or different stage of drug product development, where when we say like these are the different type of clinical study in the phase 3 we company used to conduct to prove that drug is safe and effective. This is a abstract of all those. Page 1, we have these many less number of volunteers. With phase, it increases. So, and we used to conduct different type of clinical study to prove that the drug is safe, effective, and with less adverse reaction. Okay, so these are the thing. Just I wanted to give some introduction about how the drug molecule moves from phase to phase. Now, coming to the industry point of view, how we take those informations, what is our role as a formulation and process development scientist in the industry. So this is what as a formulation scientist, so your role will be like end-to-end drug product development, starting from discussing with type of API, which form of API, which if it is hydrochloride or succinate, which salt is that. Then you start discussing with it's completely from API to regulatory filing. Up to regulatory filing, a formulation scientist involves. They work together with cross-functional team, with regulatory QA, QC, quality assurance, quality control, analytical development, production. So it's a complete, from start to end. The role of formulation scientists in industry is critical, starting from selecting the API, and we'll have a discussion on these points in the later slides. So basically, the role from initial phase to end phase till regulatory filing, approval, and regular drug product approval, and responding to the any queries from the regulatory agency. So the role of formulation scientist is critical over here. So now we saw like previous slides, there are different type of drug product development, and as per the regulatory norms, we have different type of drug product development. So we have taken examples for the development of ANDA on ANDA. So when we say a abbreviated new drug application, that means already the NDA is existing in the market. And why we go for generic, it's more related to the cost. So cost, when a generic product comes to market, definitely that has a cost impact, and it is accessible to every individual with a lower cost. So to start with the generic product development, when a generic product comes for a development, like we suppose we're planning to develop a product which it's suppose any R&D is existing in the market. So when we start developing a product, we start with characterization of reference listed drug. So when we say that these are the few points what we consider during the characterization of reference listed drug. This is the brand product. Here we see like clinical aspect of the drug. What is the route of administration? What type of dosage form? What is the pharmacokinetic ADME of the drug? So what is the drug release? We take that drug and we analyze for the official media. If it is in monograph, then we'll go for monograph drug release profiling. If it is not in monograph, so we used to see like if suppose the drug dissolution media is available in the OGD or not, official generic drug for the in dissolution database from the FDA sites. If suppose monograph is not there and OGD also not there, so we have to characterize, we have to study that different solubility, multimedia solubility. Then we used to see that PK of the drug, what is the ionization constant of the drug, what would be the, depending upon the PK, what will be the site of absorption of the drug. Then we decide on the drug release by seeing the sink condition. So these are the things. If the drug dissolution media is not available in monograph or OGD. Then physiochemical characterization. So here in the as a formulation scientist, we see like if suppose for example we'll take one example here. If suppose the physiochemical characterization, here we have to match. We have different guidelines for with respect to size and safe. Suppose a product is existing in a market with a tablet with lesser than 17 mm dimension. So what would be the dimension? Normally, generic market will prefer to match the drug product size and shape. Sometimes there are few challenges there. If the drug product, there is a chance that if suppose it is going above 17 mm, there is a restriction. We cannot change the size and shape of the drug product or tablet. So during the characterization, we do the literature search by seeing different site. And there are, if you see the drug product label, there will be always a disclosure of active and inactives, and there will be a clue saying that if suppose there is purified water is there and HPMC is there, so normally we can guess okay, this will be a weight conversion process. So those type of clues will be present in the on the label of the drug product. Then we have to study that if is there any information about the drug substance, is there anything mentioned over there, which short, is there any specific drug substance particle size is required, which class of the drug, it's a class 1, which is class 1, class 2. So we have to see and characterize the drug substance. Yeah, this point we discussed. So we used to see that R&D level, RLD level, saying that what is that information available on the label, what type of dosage form, what is the dimension, what is the color, is there any functional coding or non-functional coding, and what is the intended population. So suppose one drug is pediatric dosage form, so we have to see those informations on the from the label. These are the common sites we used to see, like for drug product orange book, where we can see the approved drug product and therapeutic equivalence of the drug. We can have all the informations like I was discussing about dissolution method database from the every site, then all the informations and patient information leaflet from the sites. Yeah, this is the dissolution method that we can see from the dissolution database. What is the dissolution method? Okay, so I have two examples here, which I've taken from ABS sites. So whenever we go for developing or starting it from the initial phase of the drug product development, so as I was saying, like we have to see that what is the quality target product profile. When we characterize, we have to see that while doing the characterization, we'll come to know what is the quality target product profile should be for the ANDA drugs. So how we'll come to know? We'll come to know, suppose we have taken the physical attributes, the appearance, we have to match color and shape with the brand product, and order size similar to the RLD. Whether there is a score on the tablet, it may be a functional score, or sometimes we used to break the tablet into two parts and we used to dose. So the score is will be scoring means the middle line where we can divide or break the tablet and we can dose. Then all physical property. This is one of the example. While when we define when we say that we are targeting or selecting quality target product profile, the drug molecule. So this is one of the chemical property we have taken. Suppose the drug is much sensitive, and we can also whether that these quality attributes of the drug product can be considered as a QTPP. So we have to say that okay, water content for this drug product may not be a CQ, critical quality attributes. So those type of informations and initial target we can get from the after characterizing the reference listed drug. So this is how the flow goes development. So from that quality target product profile, we have to select that quality attributes. From that quality attributes, which are the quality attributes from among all those which are the critical quality attributes for the particular type of dosage form or ANDA. So when we go for the targeting a pharmaceutical equivalence product or bioequivalent product, so we have to see from these quality attributes what are the parameters. Suppose it's a low drug compound, the drug load is only 3% in the in the tablet. So content uniformity will be critical quality attributes here. And if the drug is low soluble, low permeable like gaseous class 4 molecule, then the dissolution profile will be as critical for this one. So among the when we characterize the RLD, we will have the quality target product profile. From that, we have to select which are the critical quality attributes we should target for that particular ANDS. If the drug product is thermally it is temperature sensitive or it is degrading with a high temperature. So degradation product will be a critical in that drug product development. So we have to consider those parameters for during the development as critical quality attributes. So when we define we discuss we have to go for that. We have to select that dosage form. We have to match with brand product. If the dose, what is the dose design? Is it a IR dosage form or ER dosage form, or it's coated or it's bilayer? So those design will help after characterizing the brand product. So route of administration, as we are discussing, so we have to match oral route of administration. If the brand product is oral, then we have to go for oral. If suppose we are changing anything, then we have to file that product in the hybrid NDA, that is 505(b)(2). Then we have to match definitely with the strength of the brand product, and these are the parameters we have to characterize at the initial phase of development, considering and the brand product label and what are the initial physiochemical characterization of the brand product. So identifying those parameters, drug product quality attributes, like we have to select like these are the parameter where from these we have to select that CQS, like if the assay is critical here, if the product is degrading with acid, or it is moisture sensitive with the degrading with respect to in presence of moisture, then we have to see whether assay is critical, degradation product is critical. So all these we have to include as a CQA of the product, critical quality attributes of the product. Yeah, if there is a chance if during the it will be sensitive to moisture, if there is a microbial growth on storage, then we have to consider this as a critical quality attributes. So we'll see like we have taken a example here just to understand like what will be the impact of drug substance on drug product quality. So how the characterization of a drug substance is important for a drug product development. Yeah. So suppose this is one of the example I've taken. I know if the biopharmaceutical classification of the drug molecule is BCS class 2 drug, so what would be the impact of the drug substance in the final drug product critical quality attributes? So drug substance characterization or solid-state characterization of drug substance is most critical because that will impact final product quality. So knowing the drug substance better, the drug product quality will be uh, more equivalent or closer to the brand or better than brand. So knowing the drug substance quality attributes means studying the solid state form, whether the drug substance has polymer, b.pharm polymer. So what is the stable polymer? If the polymer is stable with temperature or with if suppose we are going with for suppose for example one drug is having three polymorph and one polymorph is changing with heating above 40 degree, then whether it is advisable to select a process or not with that as a weight conversion process, or whether we can dry those granules above 40 degrees. So all those parameters affecting process, manufacturing process, all will depend on these drug substance properties like solid state form, particle size. Particle size one of the parameter where it has a direct impact on the if the molecule is what would be the better what is the impact of particle size, different particle size D90 of the drug substance on the dissolution profile, metro profile. So in the same way, what would be the impact in the of the particle size on in vivo? So definitely it is a common thing, like we know that small lower particle size will definitely help in the dissolution in vitro and in vivo profile. But at the same time, how it is relevant for the manufacturing? If the drug load in the tablet, suppose it is 5% only, then it may not impact. When it goes more than 20% or 30%, so how it is going to impact the manufacturing process? So there, particle size plays a critical uh, while designing the process. Hygroscopicity, yes. If it is hygroscopic, then it will be a challenge with respect to stability of the products. That may be a chance where the degradation will be higher. So the shelf life of the drug product will go down. The product will degrade faster at different stability condition. So that hygroscopicity, solubility of the molecule, like we say, like when we the solubility, if it is viscous class 2 drug, this is one of the example only I've taken. So if it is a class 2 drug, then how it is going to impact. So the impact on the dissolution will be higher, and for the absorption, it will be a rate limiting step for the viability of the drug substance. Yeah, moisture content is again another. When we interact with a different vendor of, or we can say like selecting different grade of API, these are the parameter we used to recommend, or we used to request the API vendor saying that uh, we need this particle size range. So there are multiple reasons for that. Like if suppose we are considering, okay, we are developing a product with to 180, 150, D90, 150 micron D90, then the drug product or we are not achieving the resolution profile, we can go ahead and we can ask for lower particle size uh, or micronized API from the vendor. Same way, the higher moisture content drug molecule may degrade the drug substance during the process manufacturing, or it will restrict us, deciding the which manufacturing process we should select. And process impurities, chemical stabilities, and physical properties, flow properties, and density of the molecule, all those are critical while selecting a drug substance, which is critical while designing a manufacturing process. So this is one of the one example we have taken. You can see this one. This is one of the molecules where what we face is the drug substance is lumpy in nature. So we can say directly like, okay, we can pass through a mesh and we can still go and process the drug substance for the drug product development. However, while doing so, what we observed is, this is one of the case study taken from my PhD work. So here, what the tendency of the drug substance is not only lumpy. Once it is exposed to room temperature, a normal condition, normal storage CRT control room temperature, so the drug substance becomes lumpy. Once you, sometimes it becomes very hard for the processing. So it creates a challenge for the drug product development. So so this is the drug substance nature, and the if the drug substance quantity is less, it may not create any challenge during the manufacturing process. But if the substance, the drug load is very high, then it will create a challenge, because initial phase you can keep and you can process the you know um, you can design the process where you may get high excellent flow property and all. On storage, maybe if suppose you are holding the drug for some time, maybe for 1 week or 2 week or 15 days or 1 month, then there is a chance of formation of lumps or ball-like structure in the granules. So that will trouble, or that will be a challenging for the manufacturing process. Again, we may require to reprocess the drug, the granules, which is not feasible, or you can say that the process is not robust. So we have to study the solid state property, like we discussed before. Like we should know that what is that solid state property of the, what is the particle size of this molecule, what is the why the lumps are forming. Is it hygroscopic, or it is when exposing to the normal control room temperature, whether it is forming lumps, what is the reason why it is forming the lumps. So what would be the impact on the drug product CQAs. So now here we can correlate with the previous slide, saying that what will be the drug product, how it is going to impact the drug product CQAs. So now these lumps are there. I suppose I have the drug load of this, the dosage form is 5% only. Now there will be a challenge where the distribution of API will be a challenge. So when deciding the drug product critical quality attributes, so content uniformity will be a critical one, where which one we should observe. We should observe throughout the product life cycle, and also if it is again it is uh, since it is absorbing moisture and it is forming lumps, so there may be a chance where it will in the tablet it will absorb moisture. uh, there is a chance of quality control failure, or there may be a chance of increase of degradation product during the storage or exposed to the different stability condition. So those are the parameters we should know all these characteristics, starting from physical characteristics like particle size, volume of melting point, solubility, and the hygroscopicity, all these parameters, what would be the impact on the finished products? So there may be a chance, like as we were saying, like impact on the what will be the impact of the drug substance on the manufacturing process. So we cannot, if say the drug is moisture sensitive, or we cannot select the weight condition process, right? So if suppose the drug is temperature sensitive, then we cannot dry this drug product. If the moisture, if the drug product is degrading above 40 degree, we cannot go and dry these granules with the weight condition process. So those are the few things we should know when we design a process from the scratch, saying that you should know about your drug substance. Yeah. So the same drug molecule we try to see in the microscope. Also, we confirm that this is forming a lumpy mass, and this is confirmed through this microscopic image. And from the lumpy mass, when we say like the drug substance has a tendency to form lumps, so definitely there will be a future challenge. So predicting the future challenge is most important. Understanding the what is going to maybe in future, like this drug will impact directly the quality of the drug product. So we few things we have to see, like as we say, like physical property and chemical property. Chemical property also we need to observe, like is there any change in volume of while storage, on temperature, or while doing the compression activity, is there any change in volume of, is there any pattern we see in the XRD. So those steps we have to evaluate while doing the initial evaluation. So physically also we, when we uh, suppose the drug load is very high, and the manufacturing process we are going to select is direct compression, then drug load is very high. Then what is the compressibility of drug individually? So if the drug we can compress with certain weight of tablet, we'll come to know, like if the drug is, we have to take the drug 80%, so what will be the impact while manufacturing a tablet? So since the drug substance has a tendency to form bigger lumps on storage, and hence it will create a challenge while doing the processing. Microscopy confirms that. Then microscopy says that it's a lumpy mass, so definitely the tendency will go, it will be like we'll have screening, and where we can mix the material uniformly, and we can process for the easier manufacturing process like direct compression. So that those are the property when we select a manufacturing process, and we have to see and evaluate those property of the drug substance. So this is what we were discussing. If the drug load is 10%, the physical property of the drug will have lesser impact. the impact will go up. Like if suppose we are going to 80% or 90% drug load. So the objective is to produce a pharmaceutical equivalent, safe, stable drug product. So these are the property may challenge you as a formulation and process development scientist. This is one of the example I've given uh, where we have challenge those type of challenge. So the role of formulation scientist here is to see that like if we are going ahead with 10% or 10% drug load, so which process we should go ahead with. If suppose we are going for 50% drug load, and what are the options we have? If suppose the drug product is 90%, so what are the options we have? Whether we'll go with a direct compression, or we'll go for roller compaction, or we'll go with weight conversion process. So to understand the solid state property of API is critical, and when these are the we understood that critical property of the drug substance. So what are the feasible or suitable excipients we can select, for the robustness of to get a robust product formula and process. So once we select the excipients and we select the grade of API, and we have a method like we are planning to go for direct compression or wet condition. So is those are those excipients are compatible with my drug? So we used to have drug excipient compatibility, maybe at different ratio. We can go with actual formula composition ratio because it is early stage of selecting ratio excipients. So there may be a chance like we will not have that exact ratio in place. So we can go ahead with 1:1 with actual formula composition, saying that if suppose we are using a lactose monohydrate as a diluent or MCC as a diluent, so we can have 1:1 ratio, or we can go for more. Then we can study like if the excipient is compatible with the active API or not. For the example what I have given in the previous slide, the drug is lumpy mass. So whether we can select a excipient which has more water content or more moisture content, is it advisable? No, it's not advisable. We have to go for if suppose the excipients with more water content, there may be a chance again that will form once you process, then later it will form uh, again lumps in before the manufacturing or compression activity. Yeah, manufacturing of solid orals. These are the uh, 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 process where process where we normally prefer to go for a direct blending approach, where if the drug molecule is the proposed ratio of the drug substance is higher, then we can go for direct blending process. If suppose the drug molecule is the drug load is less, or that may be a chance of continuity issue. Dry conversion may be an option, rural compaction may be an option, because only mixing may not help. So mixing along with the geometrical mixing or optimizing the mixing process will help for the low drug level. But there is a chance of risk over there. So the selection as a process development scientist, and they will go for the dry conversion process. And there are other methods are weight conversion or spray conversion. So then again, it depends on the if the drug is again sensitive to moisture, or with water it degrades. Then we have to see that process whether it is feasible or not. Once we select this process, and we selected this one of the process where we say like weight conversion will help in this process. So then we have to go for the compatibility study where we go for both dry and wet condition where we have to mimic the actual manufacturing process, and we have to say that the drug substance is stable with respect to manufacturing process. Yeah, so this is one of the example where when I was saying like the drug load is less, so there is a chance of uh, better mixing with respect to this is a roller compactor, and when we go for uh, dry granulation, we use this compactor for the preparation of flakes. Then we needs to follow like since we have different screening step, we used to prepare the pre-RC blend by before loading into the hopper of the roller compactor, and we have a screening step. If the drug load is very less, suppose it is 3%, 5%, there is a chance of better mixing over here. Like we have screening step, we have pre compaction preparation of pre-RC blend where we will have to mix the blend. Then again we are shifting and we are compacting, and the blend and producing flakes. And then we have as we can select the different cycle number of cycle also with a different roller pressure. We can optimize those process requiring based on our requirement to attend or to get a quality products. So RC blend, it is like it goes through these steps. We used to uh, screen those material. Then we used to prepare the pre-RC blend by mixing those blends, and we used to have roller compaction producing the uh, flakes. Then we can break those flakes and pass through those flakes to different mesh size to attain a different granules required granule size. Also, we can go for cycle 1, cycle 2 based on our requirement, and then we can proceed for the compression activities as per the our requirement. Yeah, once this, what we saw is like once we studied the API well, we can select the manufacturing process. We can select the excipients. We can select the manufacturing process. We can develop that process, and then we go with uh, for normal steps. Like once the manufacturing process is developed, then we covered all those excipients. Then we did the process pre-formulation study, compatibility study. Then we challenge the process. We can optimize the process or we evaluate the process. Then we do that initial risk assessment with these drug substance and excipients. Once this process, suppose the drug load is low, and the process we share like there is a chance of failure with respect to, or there is a risk with respect to content uniformity, then we go and do that process challenge study where we have to optimize the number of mixing revolutions, and then we evaluate those in terms of uh, content uniformity. So we have different guidelines. So ISP guidelines and recently published paper, like we have to go for statutory content uniformity with a minimum of 20 locations. So once that is confirmed and results are satisfactory, then we have to update the risk saying that okay, the risk is nullified with this number of revolutions. Suppose we do evaluating 10 minute, 15 minute, and 20 minute, we have a better result at at 15 minutes, then we can say 15 minutes is optimum, and the operating range we can give with respect to 15 minutes as optimum time. Then we develop and finalize the development with qualitative and quantitative composition. Simultaneously in parallel, we go for analytical method development. When we say like these are the parameters to be evaluated in vitro. So these are the parameters. When we in the second or third slide we discuss what are the critical quality attributes with respect to ANDA development. So suppose it's a class 2 molecule or class 4 molecule, so resolution definitely will be a challenge. So for that, we have to develop method for different in vitro study like resolution assay. So for class 2 molecule, definitely it will be a challenge. So we have to see that development process of resolution method. Then finally, we come up with a lab scale stability study where we have to prove that with this final composition and final qualitative and quantitative composition, the drug product is stable. So then we can go for the scaling of the process with all these. So when the CQA is established, we do the initial study with the final composition. Then we go and increase the scale from the lab scale to uh, production scale at different batch size. Then we develop the analytical. We try to prove that the method is working for the high scale batches, or the high scale batches is not showing any kind of change with respect to the method. So we finalize the analytical method and we go for uh, lab scale to uh, larger scale after mitigating the risk uh, at the different stage of manufacturing. Then we go for scale up for the process optimization. Then process challenge study we do, like finding out the operating range for the drug product manufacturing. And then we go for exhibit batch manufacturing with final process and with all control strategy. And then we go for the regulatory filing with those three exhibit batches. So learning from the slide is today's discussion is more like so understand the manufacturing, understanding the physics and chemistry of the drug substance, and see like what is the percentage, what is the nature of the drug substance. So physically, with respect to flow, all those solubility, melting point, all those physical properties. What is the surface morphology of the drug? What is the XID of the drug? And also know that chemical part of the drug substance before designing a manufacturing process. So understanding the drug substance property will definitely will be helpful developing a robust manufacturing process. And uh, it's not like developing a process or a product. So once it is filed, it's not over. So we have to see about the life cycle management of the product. We have to find out the control strategy of the drug product manufacturing, what is, and we have to observe continuously through process validation, like how the product behaves starting from exhibit batch uh, to the commercial batch. We have to continuously observe that for the maybe for batches. Then we have to as a regulatory requirement, we have to submit the annual report for that, how the drug is behaving. So and so there is a strong belief like if suppose we are developing, we are understanding the drug substance well, and we are selecting the process as per the nature of the drug substance, and we are trying to see that whether CQA, we are meeting the CQAs as per the brand product, then we go for so once we do all those things, easy and economical process always a dream for process development scientist. Everybody wants to have that. As a process development scientist, I would like to prefer, like, first I should try with uh, direct compression or direct blending, so it will be a easier process. So sometimes uh, it is not that easy, like, selecting with a direct compression process. We have to have um, select other process, like I told before in a few previous slides. So it's all depends on how the drug substance, what is the nature, physics and chemistry of the drug nature. Sometimes we have to go for uh, roller compaction and weight conversion. That is the basic need of that molecule. So if suppose we can find out, like, we can develop a easy and economical process, so definitely it will have impact on the final cost of the medication. So uh, yeah, these are the thing I have for you. So any questions?
Thank you so much, sir.
Yeah,
We extend our heartfelt thanks to Dr. Saroj Kumar Rout for delivering such an insightful and inspiring session on drug product development and industrial approach. Dr. Rout, your expertise and detailed discussion on industrial nuances of drug product development, including formulation techniques, process optimization, and addressing challenges in manufacturing, have been truly enlightening. Your ability to bridge the gap between the academic knowledge and industrial application has provided invaluable insights to all the participants. We deeply appreciate the time and efforts you have invested in sharing your wealth of experience with us. Your talk has not only expanded our understanding but also has motivated many of us to explore innovative solution in pharmaceutical development. Thank you once again for such a remarkable presentation.
thank
So
you.
now there are some questions from the participants, and I will be asking you one by one. So the first one is,
Yes.
how does the characterization of the reference listed drug products influence the design of generic formulations?
Uh, yes, thank you for that's a question where Ajay, we as we discussed in the previous slides, so that is the base of development of drug product. So when we go for characterizing the RLD is reference listed drug. Reference means we have to refer this drug. Listed is listed in orange book. So these are the drug. This is the basic of drug product development, where when we go for under ANDA developments, so those these are the primary character we have to follow by characterizing the reference listed drug. So we have to see, uh, yeah, we have to match appearance, order, size, and all the physical property. Also,
Right.
definitely the identification, like, content of the drug, and we have to characterize. We have to understand the drug from the RLD from its level, and saying that what is the criticality will be for the drug product development. So yes, so brand product characterization is the initial and first step of the drug product development, where we see, like, what is that? Suppose we are going for a pediatric development. Yes, so we should know that what is the target population, or no intended population. It should be a pediatric patient. So we should characterize it. All those information we'll get from the brand product.
Right, sir. Thank you so much, sir. The next one is,
Yeah.
sir, how is risk assessment integrated into the formulation and process development, and how is it updated during the scale up?
Yeah, so as we have, I think I have given one example. The risk assessment, when we do the risk assessment, we do that initial risk assessment with respect to a drug product. Like if the drug load here it is 10%, the risk will be a challenge with respect to with respect to drug load, the content uniformity of the drug. So that from the initial step, we will be knowing that yes, the drug load is 10%, so it is lower than 25% of recommended. Where we have to go for the content uniformity testing, where that there will be a challenge with respect to content uniformity. If the drug is micronized drug, yes, there is another chance of distributing the drug throughout the blend uniformity. Uniformly distributing the drug in the blend will be a challenging. So definitely that will impact the you know uh, uh, final drug product quality or process. So we have to optimize, so we have to select, okay, what is that parameter which will impact, what are the process parameter which will impact the final content uniformity. So we have to select the blending process or which manufacturing process will mitigate this risk.
Right, sir. Thank you, sir. The last one is, sir,
Mhm.
how can bridging the gap between academia and industry accelerate the innovation in drug product development?
Yeah, that's a very good question, and it's a initiative has to be taken from both
Right, sir.
industry and university. So more and more the industry people will come close to the students, like through SPSR or through. So those are the frequent interaction, and also industry funded project in the university also will encourage basic research, and that will create interest in the students' mind. So they will come to know like what it exactly goes. So this example, whatever I have given, this may be a small example, but this will
Right.
be a definitely 1 month work, some work for the process development scientists. So definitely the collaboration from both sides will definitely help.
Right, sir. Thank you so much, Dr. Rao, for answering all the questions with such clarity and depth. Your thoughtful and detailed responses have provided invaluable insights into the intricate process and challenges of industrial drug product development. We truly appreciate the time and effort you took to address every query, making this session highly engaging and informative. Your expertise has been incredibly inspiring, and we are grateful for the knowledge you have shared with us today. Thank you once again for your remarkable contribution to our 77th SPSR International webinar. So here we come to the conclusion of this session. As we conclude our 77th SPSR International webinar on drug product development and industrial approach, we extend our heartfelt gratitude to Dr. Saroj Kumar Rout for delivering an exceptional and insightful session. Dr. Rao's comprehensive discussion on the phases of drug product development, critical quality attributes, formulation strategies, and manufacturing processes has provided us with a wealth of knowledge and practical insights into the industrial perspective of pharmaceutical development. This session has highlighted the importance of robust formulation design, process optimization, and regulatory compliance in delivering safe, effective, and high quality drug products. It has also reinforced the significance of bridging the gap between academia and industry to drive innovation and advance pharmaceutical science. We sincerely thank all the participants for their active engagement and thought provoking questions which enrich the discussion. Your enthusiasm and curiosity drive the success of these webinars. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collaborative efforts, which have broadened and our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We look forward to your participation in future SPSR webinars as we continue to explore groundbreaking topics and foster knowledge sharing in pharmaceutical sciences. Thank you once again, and we wish you all the success in your endeavors. Have a wonderful day ahead. Thank you so much, sir. Thank you from Team SPSR.
Thank you so much. Looking forward to future collaboration with students, and thank you so much, Sachi Alok sir and Dr. Pradeep.
Thank you so much, sir.